Thank you so much. Okay, let I share my screen.
Okay.
Sorry, just a moment. Because I used two screens. Okay. I hope you can see my slide, right?
Yeah, that's clear.
Okay. Okay, so actually, I would like to thank the Yemeni Students Union in China and Mr. Yusuf Akabi, my ex-student. Thank you so much for today's invitations for the seminar in the Introduction to Biomedical Engineering. So actually, I gave the topic to Yusuf that maybe I would like to share, not just like a lecture, because I would like to share my career path, I think, because I have a background in mechanical engineering and why I become like a biomedical engineer. Okay, so actually 30 years ago, I think, because this is my experience in Thailand, normally students who would like to be the engineer, they did not like biology. I think, some of you may like this also. Because we think it's quite totally different, from the engineering or the biology. But now, I think everyone maybe know that, we cannot like avoid for this. If you are the biomedical engineering, so you cannot avoid for the biology or medicine. So I will tell you the story. This is my educational and working route. Actually, I graduated like a long time ago. Like in 1996. In mechanical engineering from Chulalongkorn University in Thailand. And then I become a mechanical engineer in like in the power plant, the petrochemical companies, in Thailand. And then I would like to go abroad for the study. So I applied to the mechanical engineering department in Japan at the University of Tokyo, for 2 years. And then I came back to Thailand and worked as a lecturer, just like a couple of years. And then, I went to US for my bioengineering study. Okay, so if I ask you that if I say about mechanical engineering, what are you thinking about? I think some of you may think like, about the engines, about the pumps, even like the power plant, right? Because it's mechanic. Absolutely. Everyone would like to think about that. And I believe that if some of you are mechanical engineers, you may learn these subjects in the class, like a thermodynamics, mechanics of solid, or even like a fluid mechanics or design and calculus. I believe this is the principle of the subject that engineers should learn. And when I was the undergrad student, the work that I did is about the vibration analysis. So I learned about like how to analyze the signal from the machines and try to find what's the failure modes of the machine, and then try to do some kind like a preventive maintenance for the machines. That is story about the mechanical engineers to do. And when I took my master degree, I think maybe I still have the field in mechanical engineering, and I like the subject about thermodynamics, heat transfer. So when I apply my master degree, I seek for the professor who can supervise me in heat transfer subject. So my thesis is about like how to study about the behavior of boiling. So you can see on the screen that I study about the the boiling and try to see the cavity arrangement design, how I can facilitate the cooling, in the boiling phenomena. And in that period, I also learn the new things like C++, the computer software, and MATLAB in order to do some kind of the calculations. So when I finished my master, I went back to Thailand, and I have to find a job actually, because that is maybe the thing that I would like to do. But I would like to go back to my hometown, so my city in Songkhla in the south of Thailand. At that time, I think, that's the question, which one should I choose? Because I apply mechanical engineering department, but they said, they cannot accept me if I just graduate only the master degree. So I applied to Faculty of Medicine. They just opened the new, the new department, like a biomedical engineering. Okay, so maybe I can apply the biomedical engineering. But my first challenge is my boss. He asked me that you should go to study PhD in biomedical engineering, because this is the new field in Thailand. So you have to learn bio and medical. As you can see in the slide, that biomedical is come from bio and medicals. So finally, I have to face with the biology, the thing that's the engineer doesn't like it. So this is my first challenge, because I forgot everything about the biology for like 6-7 years, I think. But when I think, what is the biomedical engineering? There are so many broad spectrum of the subject in biomedical engineering, like biomechanics, robotics, bioinformatics, or even like molecular biology that you can apply for the molecular engineering. So what should I do? Because I have a background in mechanical engineering, right? So I have to think that, okay, maybe the computer is the thing that maybe I can try, because I have some background in computational a little bit. So I start with my thesis about the cardiac simulation when I was in US, but unfortunately, I could not stay with my ex-professor, because I cannot reach the level that he expects. So I have to change my topics, switching from the computer to animal study. So this is my second challenge in my life, I think, because as you know that I work with machine, I work with maybe the non-living things, as an engineer. So how can I handle the animals in order to study? But I have to try. So that is the change that I had to study about cardiac function in animal. So you can see that this is my animal that I use in the study. They are hamsters, because I have to put the catheter inside the left ventricular chambers and try to see how the heart works. By using the software that you use, you can analyze this one, how the functions of the cardiac performs. That is really challenge to me. That's I have to know about the surgery. I have to to try the drugs or some kind of the plasma expander that never know what is it at that time. But anyway, I think I learned so many things when I was PhD student, because the first one is the research is sometimes cannot control, and there are some uncertainties, because I work with the animals, and at the beginning my animals, they are dead. You can see that's 20 animals dead and just survive only 25 animals. So that means nothing is smooth, but I learned from the mistake. So at the end, I think I can handle the animals more than 80% that I can make them survive. And also the other thing that I learned is how to manage my project, because you have to do the progress report to the advisor. So I think manage good is make our life easier. So yeah, that is what I I learned. And also my friends, yeah, he taught me about to think outside the box, because this one I think is really important for engineer, because as I told you that at the beginning, my animals, they are dead. And my friend asked me to look at the result, why the animal dead. Yeah. So yeah, just analyze the data, and maybe you can figure out one paper from that failed work. Yeah, because everyone may think like, yeah, it's not success, and maybe the data is useless. But yeah, I got one paper from that mistake. Yeah, and you can see that this one is like a functional recovery after hemorrhagic shock, I think. So yeah, this is my first paper that I published. Yeah, from my friends who initiate the idea, think outside the box. You can do it. Oh, sorry. I think. Okay, and then my advisor, yeah, before I graduate, my advisor, he told me that, yeah, when you go back, don't don't forget to publish. He asked me to keep publishing. Yeah, even like yeah, you don't have any like a interesting thing, but this is the skill that you have to practice, and if you can keep publishing, maybe for your career path, it will be benefit. So okay, that's is my advisor before I go back to Thailand. So it's the third challenge to me, because yeah, I have to come back to Thailand and work as like in the academic fields. So what should I do? I asked my question that what should I do? So maybe I can start from my interest and what I have for the skill, and what the resource in Thailand that I can use. So yeah, maybe engineering is the thing that I have to think about, because I think I learned from the engineering that's maybe about the problem solving, systematic thinking, creative thinking, mathematics, and hand-on skill. I think these are the thing that I learned when I was engineer. So I think I can do it. Even like nothing in Thailand, like not the same as I did in US, but I had to start my career path. So I came back, and then I set up the work about the cardiovascular engineering, because as my background is engineering, and I work about the cardiovascular when I was PhD student. So yeah, I think this is the thing that I have to try when I came back. So during the first 5 years when I came back, yeah, I worked on the cardiovascular engineering, and I also did in vitro study, in vivo study, and in silico study. So I can tell you in some detail, but not all, because I think the time is not much. So for the in vitro study, I look at the blood. You can see that because my PhD work, I study about the plasma expander, which I have to substitute the blood. So I have to look at how the red blood cell change when it interact with my plasma expander. So yeah, you can see that I look at the control. This is the RBC shape. And when I mix it with plasma expander, it's like the plasma expander is a hydroxypropyl starch. Yeah, when I put it at the room temperature for 4 weeks, you can see the shape is a little bit changed. But when I increase the concentrations of the plasma expander, you can see that the red cell change a lot. And also when I increase more, the shape of the red cell is totally different from the beginning. And after that, I have to put this plasma expander into the animals in order to see the systemic response. So I use the window chamber models in order to look at the vascular system. You can see on the slide that we can see by eyes that how the vascular network is. And also, we have to look in the detail in the microscopic level. So you can see the blood flow inside, and then I have to analyze how my plasma expander change the vascular system, like a diameter, the flow, something like that, and the capillary density of the animals. So the model is this one. You can see that I have to do the surgery on the animal and put the window chamber on the back, on the dorsal of the hamster, in order to see the detail of the vascular system. And then the other works that I just start, I think last 5 years, is about like the in silico or the computational modeling. This work it was done by Yusuf when he was studying here in Thailand. Because I think as engineer, maybe the computational, maybe the good thing, because it can save money a lot, because you don't need to deal with the chemicals, or you don't need to deal with the animals, because animal study now has the regulations very strict. So when you were to use animal, you have to get the approval from the ethics committees, and it costs a lot. So I think using the computer for the modeling maybe is another way that I can do the study about the cardiovascular engineering. So these are like my research work right now. And last 5 years, I think I can still keep the cardiovascular engineering, but I have to expand a little bit more on in order to make the impact to the patients, because the cardiovascular engineering, as I told you, is very long run research. Because if you would like to put something inside the body, that's very long study. So maybe 10 years, maybe you get success in the first step of the clinical trial. That is a little bit challenge, but I think we can get easier that patients or the people can use it if we work outside the body. So I think about the rehabilitations engineering. So we can do some kind of the training or the device, the external device for the patients. So that's why I start the new field of my research about the rehabilitations. So you can see on this slide that we developed the device that can help the patient who has a stroke. When they walk, they have some abnormal gait during their walks. So we try to use this device to feedback for the walking pattern. But it's still in the first step of research in the clinical study. And we try to use the simulations in order to see how can we make the footwear for the patient also. That is for the diabetic foot. These are my current research right now. And even about the computational modeling, I still keep on the vascular system, but focus more on the patient disease model, like aneurysm, as you can see on the slide. And also for the EMG, this is the new thing also for me. But as I told you at the beginning, my mechanical engineering background, I deal with the signal analysis on the vibration. So I think, EMG is also the signal from the body. So if we apply the knowledge about the signal analysis, maybe it's useful yeah, for this field of the rehabilitation also. Yep. Okay, that's my current research and my challenge during my switching from mechanical engineering to biomedical engineering. And as Yusuf told me that maybe I can like introduce about the field of biomedical engineering in Thailand or at PSU, my university. So not just only the cardiovascular engineering that we have, but we have like 5 core technology in PSU biomedical engineering. So we have the group about the biomaterials and regenerative medicine. This is one field in biomedical engineering that develop the new material for replace the organs such like a bones or nerve, something like that. And the other group is about the biosensor and the lab on the chip. So this one is really high throughput technology, I think, because yeah, you may know that the diagnostic kits during the COVID is really important. So yeah, this field they can develop the biosensor that can use the small amount of the blood and they can diagnose what's inside the blood and you have any disease or not by using that kind of technology. And the third one is about the biomechanics and medical devices. Yeah, this one is the field that I told you already that yeah include like the motion, the gait, yeah, some kind of the cardiovascular engineering also is in this group. And the fourth group is about the molecular medicine and nanomedicine. Yeah, this one is also become like the new technology because as you can see that right now we try to have some kind of patient-specific, right, or we call precision medicine. So you can develop the new drug that's targeted to the specific site, especially for the cancer therapy. Yeah, you can see that we would like to avoid a systemic, yeah, like effect. Maybe, yeah, we just target the drug to the point that has the disease. This is will be like the new treatment in the future for the targeted therapy, something like that. And the last one of our technology at PSU is about the AI or medical data analytics. Yeah, no one cannot disagree that this era is become like an AI era. Yeah, everything that you look at, you say, yeah, in the future maybe AI can do that also. And I think for our 5 group, the final aim that we would like to do is we work the research and we try to implement or we try to apply our research to the bedside in order to make like the patients to be like get the wellness. Yeah, that is our like mission for the biomedical engineering at PSU. So if you would like to see in detail of our like research technology at PSU, you can scan the QR code in order to see the video that we have explained what's PSU biomedical engineering is. And also I would like to get the announce or maybe some kind of the promotions. We also have the scholarship for the international student to study at the Faculty of Medicine. Yeah, we have like a four scholarship, but for the biomedical engineering, we have just only 1 scholarship per year. Yeah. So these are the schedule of last applications. So if you would like to apply for next academic years, maybe in January to April of next year, we will open the call or the applications for graduate study. So we have like several international students in the programs. Yeah, you can see that there are 4 right now, but actually we have 6. Yeah, but I could not find their pictures. Yeah, one of them is Yusuf. And I think this is my last slide. Yeah, this is my group. Actually, as I told you that I'm graduate for a long time, I supervise the student about like 20 students for my master student and my PhD student that I supervise. Yeah, are listed here. And thank you so much. I think I spent a little bit on my like a research and career and the biomedical engineering at PSU. So I think we still have time for the Q&A, right? Yep. Thank you so much.
Thank you, Doctor. I think the next Dr. Rina.
Yeah, sure.
Okay, I will make you co-host.
Here are question and answers, brother, Mr. Adil. Maybe we can leave the questions and answer if someone want to ask.
It's okay.
Yes.
May I ask?
Hello, Shaubi.
Go ahead.
First of all, thanks, Doctor, for giving us some of your time. But my time is the same as you said at the first. You said you were like bachelor students for mechanical, then you changed your major to biomedical. Normally, for mechanical students, they are more concentrated with setability and maybe machines, how to derive, design, something like that. But for biomedical is more about medicine, biology, so it's totally different major. So how could you manage this thing?
Yes, I think it's very good questions because it's totally different. I agree that, but I believe that we can learn the new things because yeah, as we are engineer, right? So I think the problem solving, the systematic thinking, something like this, can help you to study the new things. And yeah, because I think it's not quite difficult if you try to like understand the new file, like maybe about the physiology, right? Like that.
For the basic knowledge, you need to continue your research, like basic knowledge about biology, chemicals, and so on. It's totally different about mechanical because, you know, we are more about theoretical, maybe thermodynamics, something like that.
Yep. Yeah, as I mentioned, because I think it's about the mindset. If you think it's really difficult to study or to learn, I think it's a barrier. You cannot jump out from the comfort zone of the mechanical engineering to the others. I think that is the first thing you have to have the mindset that everything can learn. Yeah, so you can learn about the cell, what cell is, or the physiology, or some kind of anatomies or chemistry. Because when I switched to bioengineering, when I was PhD student, I have to take a class. I have to take a class on physiology, on the biochemistry, even like a tissue engineering. Yeah, and I have friends who can help me during my study. But when you get involved for the research, that is the period that you have to spend a lot of effort, yeah, to learn, because that is no one tell you about the research in biomedical engineering. You have to self-study. Yep.
Okay, thanks. But actually nowadays a lot of students, they are switching like you. He was like mechanical or other majors, then they changed to biomedical. I just asked some of them, they say the key point for this is easier, or maybe now a lot of people are searching with this area. So thanks for that.
Sorry, if no one asking, I have a simple question, Dr. Swabo. Do you advise young researchers to switch majors, like if they are like, if they feel like they are not any more interested in their bachelor degree courses, and they want to impress their future? Is it a nice step to do, or sometimes it's risky, challenging, or something like that?
I would like to say it's a challenge, step, because as I mentioned, the biomedical engineering is a new field, right? So I think, and you may see that during the COVID situations, many equipments or many devices, are invented, I think. So this also like the challenging, for the people who like to switch from mechanical engineering to biomedical engineering. Yep. I'm not sure that I answered your questions or not, Yusuf, but I think it's a challenging,
Right. I guess maybe like could be an answer for everybody here who is like attending. Thank you, Supervisor. Chancellor. Adil, I think you can go ahead.
Okay, maybe my internet unstable. Dr. Rina, you can start, please.
Still, we have one question here.
Where? Jamil, you can open your mic.
Hey, hello everyone. Hello, Professor. I have one question.
Yep.
Can you hear me?
You can ask. Yeah.
My question, is there a chance, now I'm doing my, I study medicine as bachelor. Is there any chance to just to study biomedical engineering as a master degree later?
Yes, because right now we also have the medical doctors who already finished their undergraduate level and they apply for the biomedical engineering at my program, I think. Yeah, and also in Thailand right now they have like several MD, MS, I think, that they combine with the biomedical engineering program also. Yeah, they provide 7 years for this track. I think you can earn MD and then the master degree, in biomedical engineering. Yep. Now there are a lot of like a program that accept the medical student to take the master degree or even like a PhD. Yep.
Thank you so much, Professor. So there is a chance to specialize later in biomedical engineering for doctors.
Yes, because now I think we try to encourage the medical doctor to have PhD also in order to like they can have both sides, the user and inventor. We try to encourage them.
Okay, got it. Thank you so much.
Yep, you're welcome.
No more questions, I think.
Yeah, maybe later. I think it's okay. Maybe I don't want to make the times. I take a time.
Okay. So I think Adil can the host. Adil, can you hear us? Maybe he got some internet connection.
Hello. You hear me, please?
Yeah, I can hear you.
Yeah, Dr. Rina, the mic for you, please. You can start.
Okay, sure. Thank you. Thank you, moderator. I will share my screen first. Can you turn on my, it's disabled actually, my share screen. Can you yeah.
Okay, no.
Yeah.
Now it's okay.
Yeah.
Dr. Rina, now you co-host, you can share your slide.
Yeah, I can access it. Can you see my slides here? Is it okay?
Yes, now it's clear.
Okay. So thank you very much, dear moderator. And okay. Let me adjust my setting first. Give me 2 seconds. So a very good evening and warm welcome to everyone, and also Masa Al-Kaye, dear participants. So today's topic will be biosensors and emerging technology. So today's session will be an introduction section for biosensors, and I will be talking about what is biosensors and what are the applications of biosensors, and also how biomedical engineering can contribute for the growth of biosensors. So this is going to be a very brief introduction on biosensing. Biosensor topic. I think I'm having some problem with my slides here. I can't move it to the next one. Can you see my slides? Is it okay?
Yeah.
What's wrong with this?
Yeah, no, it's okay.
Yeah, are you seeing my bar over here? This mute, stop video, all these bars?
Yeah, the next slide.
It's blocking actually, right? It's blocking. Okay, yeah. So yeah, let me start again. So biosensors and emerging technology. So if you see here in our current world, we as human beings here, we are contacting with our surrounding around us using our sensory organs, means our ears, mouth, eyes, tongue, and our skin are the sensory organs to interact with our surrounding. How about we are not alone in this surrounding, right? We are sharing our space with invisible organisms. Usually we call it as microorganisms like bacteria, viruses, protozoans, fungi. We do have many other microorganisms sharing same space with us. So for this, with the advent of technology, we can see this using microscopes. So these are the very basic equipment, microscope. But let's think about cellular level biomolecules, for example, peptides, proteins, ions, gas molecules in environmental samples. So how we can detect or see or diagnose these things, how to detect it. In that case, there's coming this biosensor. So with the advent of nanotechnology and also biomedical engineering and also some other fields, which I will explain in our upcoming slides, we can see that biosensors is going to help us, it is helping human being in detecting all these cellular level biomolecules. So biosensors are very handy, easy to use, and also it's very important for detecting biomolecules, cellular level biomolecules. Therefore, you can see here, the word biosensor itself is a self-explanatory, means it is bio, plus with sensor means it is a sensor. It is a detecting platform, sensing platform. It comes with a bioreceptor. It means that sensing platform, it has biomolecules, bioreceptors attached to it in order to sense or detect targeted biomolecules in any samples, for example, sweat sample, saliva, blood, and environmental samples as well. So what we can detect in the samples are example, you can detect viruses in human body. For example, in this pandemic, in COVID, we are using biosensors, the rapid test kit to detect coronavirus. So that is one of the example. And following that, for diagnosis of diseases and then to detect pathogens, viruses, bacteria, salmonella, and so on. And there are a lot of examples which we will see in detail in upcoming slides. So therefore, biosensor is an analytical device, or we can call it as a chip. It is widely being produced in research labs, higher educational institutions, industries for two purposes. One is for R&D works, for research and development, and another one is commercialization work. That's why we call as this biosensing technology is an emerging technology, which means we do have sensors, but there are very few established sensors, okay? So therefore, biosensors are important for detection of biomolecules and also for detection of many other biomolecules for different various fields. So let's look at this. These are the examples of well-established biosensors currently in use. So it is helping in improving livelihood of mankind. So for example, blood glucose monitor. This is helpful for diabetes patients. So what they have to do is they just take a needle, they poke it here, and then with the prick of blood, they just put the blood over here, and then it will flow into this device, and then it will show us what is the blood glucose level in human body. So this is important for diabetic patient to monitor their blood glucose level in their body. Following that, pregnancy kit. This is one of the very well-established biosensor in our current use. So usually they will use this urine or blood samples, and then they will put it here. And there are two types, means based on urine and blood sample, then put it here, then it will show if there is double line that it's pregnant and this is not pregnant. So this is quite this is well established. And last but not least, this one, rapid test kit, RTK. I think all of us knew about it very well. So this is one of the example, shows that biosensors are important in improving livelihood of mankind, whereby it is helping in our pandemic to test, to diagnose people with COVID-19. So that's one of the major significant contribution of biosensor in current situation, RTK. So let's look at this. What are the key components of a biosensor? As I mentioned earlier, biosensor comes with a transducing channel, which is a sensing platform. Okay, this sensing platform is important to translate the binding of the binding of the target with the bioreceptor, and it has a bioreceptor on top of it. So sensing medium here, and then it is attached with this bioreceptor, this probe molecule, and then when let's say there is an input channel, so let's say when there is a blood, saliva or sweat sample or anything with some kind of viruses or pathogens like here. So what happens when we put on top of the sensing platform, let's say there is no virus, this blood is clear without any viruses or pathogen, so there is nothing will happen. But when the blood sample contains pathogens, I mean like virus or any bacteria or anything, then this binding will happen. Can you see this? This is probe, and then this is the targeted molecule. When this binding happens, then there will be some changes in the signal. Means this transducer will convert this translation, this binding event into some readable signals. So there will be some readable signals. So it can be, we can read that. So this is what, this is a simple explanation of it, how this biosensor works. So let's look at this schematic diagram again to recall you. So there is analyte, and then these are the targeted molecules, and this is transducing channel and bioreceptor. So it will bind the moment the target and the receptor binds, the transducer will change the signals into measurable signals, and this can be detected in the detector. I mean, it will display the result. For example, in COVID cases, it will display whether you have COVID or not. Okay. That are the key components. And next, let's look at the working principle of biosensor. So for this one, I took an example of one device, which is graphene-based field effect transistor. So this is a basic device. Let's look at the diagram number 1. This is the basic device. It has this transducing element is here. So the transducer here is graphene, which is a 2D nanomaterial. That means for biosensor, we also have the significant input from material science field. Okay, the idea is that. So if you see here, this is the graphene channel. When you look at the curve, this is the current IV curve. The curve is like this. The moment, this is a basic device, but the moment you want to change it into a biosensor, then you need to add this bioreceptor. Every bioreceptor is specific to certain biomolecules. Let's say if you want to detect coronavirus, COVID, then you should have a specific bioreceptor for coronavirus. So when you add this bioreceptor, look at this, there is a red color curve. It means that the curve, the current changes from this point to this point. So this shows that graphene is the transducing element, which means it can know if anything comes in contact with it, it is highly sensitive. It can translate that reaction into output. There is a signal. So let's say now we are testing with the sample. So from basic device, it becomes biosensor, and now we are testing with sample. This blood sample, it contains some sort of a virus or pathogen and anything. For this case, I'm using again, coronavirus example. So when this aptamer, when it attached with coronavirus, that target, so there is a green color curve. So this is an indication that that blood sample contains the target molecule. So binding happens, there is a change in the curve. So this is how biosensor is working actually. It can translate the binding event between the target and also the probe or bioreceptor, and it will give us the idea, it will give us the indication that something is present in this sample. So this is one of the example, which means the transducing channel is an electrical channel. There are many other channels also based on optical, colorimetry. So I don't go in detail. This is just for your basic understanding. Okay. So next is, therefore, biosensor is one of the multidisciplinary research fields. It's a product of multidisciplinary research fields. So if you look at this one functional unit comes with 5 different fields come together. It connects together for the birth of biosensing technology. For example, the transducing element, that graphene, example graphene. If you want to use it, then you should have this knowledge of material science, or any expert from material science can come in and they can help to build this biosensor. And then these probe molecules, you need the knowledge of biology. And then for transducing channel, there are some physiochemical changes happens, physics and chemistry. The knowledge of physics and chemistry is important. Finally, medical engineering or engineering field, particularly we can say nanotechnology, microelectronics, nanoelectronics, biomedical engineering. This engineering knowledge is important to make one complete functional unit of a biosensor. So biosensor is not only confined with one field, but many fields come together to produce this biosensing field. And it will be very interesting because you will learn a lot. You will learn a lot. You will learn a lot. You will gain a lot of knowledge. So if you are a biomedical engineer or biomedical engineering students, and with extra knowledge of nanotechnology, you can work on this. And always remember that in biomedical engineering, I hope that there are many students here who are biomedical background. So if you are biomedical background, you know that you'll be learning on biomaterials. You'll be learning equally, both medical subject and also engineering subject. So that's the advantage of studying biomedical engineering. So you will know about biomaterials, artificial organs, biochemistry, anatomy. So all these subjects, you can use their knowledge to produce this biosensor. So let's look at the history. So the first true or actual biosensor was produced by Clark in 1956. We call him as a father of biosensor. He is the father of biosensor. So he invented oxygen electrode, which is also known as the Clark electrode. So this electrode actually sets the benchmark for the birth of glucose detection, means glucose sensor. Okay, in 1962, then following that, They have the first potentiometric biosensor for urea detection. And finally, the first commercialized glucose biosensor was produced on 1975. And now in 2021, we have well-established glucose sensor. Not only this one particular device, but it comes with AI application where even the data can be transmitted to your smartphone. So the technology has developed very much. Okay, but this is for glucose sensor, but we still have room for improvement for other applications as well. Okay, for glucose, it is a well-established sensing platform. And so this is types of biosensors. So mainly there are 3 types of biosensors. We can categorize it into 3 types. One is electrochemical, physical, and optical. So electrochemical means this is the working principle, how the transducing channel works. And then this is for physical sensors, and this is for optical sensors. So I don't want to go in detail because this topic comes with a lot of it's a very huge topic, so I'm just giving you a brief introduction to types of biosensors. So let's look at the advantages of biosensors. Biosensors, why biosensors are very important? Because it is highly specific, number one, it's easy to use. It's independent of factors like stirring. Let's say if you're using some other methods, when you stir, the variable might be different. The temperature, pH might affect the result of the samples. But when you use the biosensor, the result will be stable and constant. So that's one of the advantages. It's a major advantage of using biosensor. Next is linear response. It is biocompatible. You can see next I'll be talking about the artificial organs. So it is biocompatible to use in human body or blood samples or in human-related things. Biocompatible. We can do on-site application, means anytime you can take out the device or a chip, you can do your testing. And easy handling, fast results, and less manpower are needed. You don't need a trained personnel to handle it. Anyone, anywhere can use it. Again, it is cheap, and also sometimes in some cases it is reusable. So these are the advantages of using biosensors. So now let's look at the application. So biosensors are not only focused on healthcare or medical system, but it is one of the, it's an emerging technology in many different fields. For example, in healthcare system, food industry, environmental applications, astronomy, in space exploration, and military applications. So let's look at the first one. So this is the application of biosensors in healthcare system or a medical field. So one of the major contribution of major application of biosensors in artificial organs. Okay, how biosensors help in artificial organs? Let's say if you want to produce an artificial organ, example kidney, artificial kidney. So you need to make sure when you produce this artificial kidney, all your function of the natural organ is restored into the artificial kidney. So how to do this? By developing, one of the method is by developing biosensors. So if you look at here, this is a picture of me. Back then I took it in 2017, I guess, when I was doing my PhD. So I'm holding this 3D printed kidney. Okay, this is a 3D printed kidney, and 3D printing is another interesting topic. Maybe we can look on it on some other day. This is another amazing technology. So this is the 3D printed, just a prototype of an artificial kidney. Okay, so what I did was, this is my PhD work. What I did was I developed an anti-diuretic hormone biosensor. So this is the chip. If you look at this chip, this is the biosensor. I made it for my PhD work. So what I tried to do was, I tried to mimic a function of natural kidney into an artificial kidney. So let's look at this. So if you see ADH, anti-diuretic hormone. So anti-diuretic hormone is very important in maintaining water balance in human body. So anti-diuretic hormone, usually it is produced at the pituitary gland of your brain, and then it will travel into your body, and then it will go into this collecting tube. Why? Because let's say there is water content in your body is low. Then this ADH will give instruction to the kidney. It will tell the kidney, hello kidney, please reabsorb back all the water. Don't let it to flow out. Means you cannot, this ADH will instruct the kidney not to produce urine, because if this kidney cannot detect this ADH, then fails to ADH, then excessive urination will happen and loss of water will happen. So these are the important function of ADH in controlling, in maintaining water balance, and also in controlling urine production. If kidney fails, then this condition is called nephrogenic diabetes insipidus. So my PhD work here is to mimic this function of kidney into this chip. Means if this chip can detect ADH, then it can be implanted into this artificial kidney, and then it will reabsorb the water. So that's the main idea about this biosensor. So next one is monitoring health conditions. So biosensors are important in monitoring health conditions, like as I said earlier, pregnancy kit, this is one of the example, blood glucose level, these two are well-established biosensors. And these are, let's look at this, blood oxygen monitors in bloodstream, monitoring heart rate, blood pressure, these are also part of the biosensors contribution. But if you see now, we have advanced level whereby we have all these smart watches where you can track your heart rate, blood pressure, sleep tracking. So this is a combination of biosensor and also our Internet of Things, AI technology, okay? So third one is for diagnosis of diseases. And again, COVID-19, why I keep on mentioning about COVID-19, because biosensors, it really makes a significant role in current pandemic. So this is the vast contribution of biosensors. So this is a promising field to venture in. And then to detect cancer cells in blood circulation, in a blood sample, but this is not a biosensor, this is a microfluidic device. So I'm just trying to, put it here for you to have an idea what is microfluidic device. And then again, emerging of infectious diseases. So it is not, biosensors is important to detect disease, to diagnose it, but at the same time, it is also important to have some safety purpose, means like we can detect earlier if there is any emerging of infectious disease are coming on. So what we can do, this work is actually going on, and what they can do is they can put this implantable chip into the animal, and then they can see if there is any chemical changes or any pathogens or any differences in the biochemistry of the in the blood of the animals. Then that is an indication of the emerging of the infectious diseases. Okay, maybe you can look into this paper. That's an interesting research work. And next is in food industry. So this is apart from biomedical, apart from medical field, now we look into food industry. So first, it is important for quality check, okay, for food safety, because sometimes food contains contaminants such as carcinogens, toxins, and pesticides. How to evaluate this? How to inspect this? Yes, by using detection kit. This is biosensor, one of the example. So they can detect all the contamination on the food. And then it is also important to detect foodborne pathogens. So biosensors plays a role over here. Following that, for food ripening and also for food quality, okay, for food quality, let's look at this diagram here. This food is packed with a chip over here. Means this chip indicates that whether the food quality is in good or it's already not in a good state. Okay, so it is to analyze its food quality and also for ripening of food. Okay, biosensors, there are biosensors for monitoring hormone levels in fruits. Actually, this hormone level ethylene is for ripening. This is the hormone responsible for ripening of fruit. And then third is fermentation process. So this is in alcohol industry, in wine production, one of the example to produce high quality wine. So biosensors, the chip will be kept there, and then they will monitor what is the quality of the fermentation process. And the moment they found the process reach an optimum level, they will stop it and then they will bring it to production level. So that's the role of biosensors in food industry. Next is in environmental samples. So as we know, in our environment, there are a lot of toxic pollutants, and one of the dangerous one is the heavy metals. Okay, so heavy metals here, it is non-biodegradable, example, lead, zinc, chromium, mercury, cadmium, and also copper. It is highly toxic and it is present in everywhere. Biosphere, it means it's present everywhere. So it might pollute our water, the water we are consuming, what we are drinking. So it is important to monitor all these heavy metals. How? With the use of biosensors. So biosensors, this is an example of the chip. Okay, so biosensors can be in the form of device or in the form of chip as well, small chip as well. So this is a nitrate sensor. So it is to detect nitrate, and also it is important. So that's the contribution of biosensors in environmental samples. So monitoring heavy metal concentration. Next is in space exploration and military application. So for space exploration, biosensors are playing its major role in monitoring health of astronaut and also crew. They are going to space actually. And then second is to monitor elements in space. If those days before this, before they come up with biosensors, those days they will bring back the samples collected from outer space. They will bring it to our Earth, and then they will process it. They will inspect it. They will do all sort of laboratory work. But now with the advent of, with the help of biosensors, they can monitor, they can inspect the sample in space itself. For example, testing of water. Okay, and following that for military. So for military, this is very interesting application. Biosensors can be used as a battlefield intelligence and significantly can counter environmental threats. Means, let's say that someone is shooting you from an unknown side, then this biosensor can detect it. So this is a very advanced level of biosensors, and in UK the project is ongoing. So you can Google about it and can read more about this as well. So how they can how they can use biosensors for military personnel. They can use it implantable sensors, or they can use patches like this, the Adh. So they can use patches of it, and then they can stick on the skin of the military personnel. Okay, so role of biosensors in managing this pandemic. So as we know, RT-PCR. This RT-PCR is the gold standard. It is highly selective and sensitive and also accurate. But the key limitations of this method, it is very expensive. So if in Malaysia, it costs us around approximately $70 USD in Malaysia. Okay, and we need a centralized laboratory, trained laboratory technicians, manpower to do this testing, and also it takes a long time, long protocols, and then we need transportation costs. And there are a lot of limitations, disadvantages in using RT-PCR. And then mainly it's about the time and also expensive, the cost. So with this RTM or rapid test kit COVID biosensors, it makes our work easy, where it is easy, fast, user-friendly, can be self-administrated at home. So if you see here, this example here, this is a COVID kit from GMAID saliva antigen test kit. So you just use your saliva, you spit on here, gently spit it, and then just like put it into they will give you a reagent, then put it into it, and then hold it put it for a few minutes, and then leave it on the sample. So this is the device actually. This is the device, and then after you put your sample over here, then it will show you the indication. So if you are someone with COVID, if for COVID patient, it will show positive. 2, C is for control line, T is for, if there is 2 double line, then it is positive COVID, and if there is only 1 line, then it is negative, and they do have invalid, means it shows that it is specific for COVID in highly sensitive as well. So what is the key advantage of using this RTK, rapid test kit? It is cheap compared to previous one. So it is just like $10, $10 USD if comparatively in Malaysia, it's 10 US dollars, and also it just take like around 5 to 10 minutes to do it at home. So that is the advantage of biosensors. Now let's look at this one. So as I mentioned earlier, my PhD work is about developing a biosensor for artificial kidney application. So what we did was, we used my PhD project, and then it has been adapted for developing COVID sensor. So this is one of a breakthrough or milestone for me actually. So before I go in detail, let's look at this biosensor first, so that you can have an idea how does it look like under this high electron microscope and all these things. So let's look at this. This is an image from camera, this DSLR, and then this is image from FESAM. And if you look at this area circled in red color, this is the transducing channel. Okay, so I used graphene. Okay, graphene 2D material. So if you look at this channel, let's look at this. This is the 2D image under optical microscope. If you look at this channel is 10 micron. Okay, what is micron? If you ever wonder, then let's take 1 mm, you cut it into 1,000 pieces, then it will produce you 1 micron. It means it is very, very small dimension. Okay, and this image below here is 3D profiler, 3D image. This is the electrode, and then this gap here is the place to put the transducing material, graphene. Your transducing channel is here, and then all the probe molecules will be placed here. So this is just to give you an idea how does a biosensor looks like. Okay. So let's look in detail. So this is the Adh biosensor. So this is a schematic representation of my sensor. So this is the device, basic device, and this is the transducing element here. And then it has a bioreceptor specific for ADH, for artificial kidney application. So what we did was, we took this basic element. So that's the key is when you do any work related to biosensors, make sure your basic device, this device is highly stable, and then it is reproducible. There is no any faulties over here. And then, so when the basic device is very strong, then you can put this bioreceptor. So it can, bioreceptor targeted for COVID molecules, and it can detect COVID. So that's the idea actually. So when you do any project related to biosensor, make sure your basic device, your electronic device or any optical device, your device is very strong, stable, no faulty, highly sensitive. So it must be an optimized sensing platform so that you can just alter the bioreceptor and you can detect any biomolecules. So that's one of it. And yeah, so our group actually, we won a gold award in COVID-19 International Innovation Award. I'm just sharing this y'all so that this will inspire you to do your work in a very diligent way, so that when there is an emergency or pandemic like this, we can use our work, our PhD work, our master work, or any research work to help the mankind. Okay. So this is our gold award, and the COVID biosensors work is ongoing actually. Okay, and this is just to show you all some images. So this is clean room environment, and I used all these equipments for fabricate the biosensor. Okay. So what is the future of biosensor technology? So this is going to be a revolution in the healthcare system. We already have telemedicine, but for future, it's going to be a revolution in healthcare system overall. So let's look at this Internet of Things. Usually we have heard of Internet of Things, but there is something called Internet of Medical Things, IOMT. It means all the medical devices applications are connected on one healthcare or network system. For example, medical devices, smart devices, smartwatches, apps in smartphones, or biosensors, all are connected. So connected means it will retrieve the data from the user, the patient, all connected on this Internet of Things, and it will be captured on cloud platform. Usually Amazon is one of the example, Amazon Web Services, and then data is stored and analyzed here. When the data is stored, it will be analyzed and then it will be sent to hospitals and also healthcare system, whereby all these medical personnel, physicians, specialists, they will see your health condition, and then they will give you medication, they may advise you. So why this revolution need to happen in a major way? Because when this IOMT is, Internet of Medical Things is well versed, so it will help outpatients, means outpatients and basic healthcare people they want to monitor their basic healthcare, they no need to go to hospitals, they can do it remotely at home. So hospitals are more focused on intensive care and also emergency cases. So that's the idea behind this IOMT, to just make our work easy and also to give priority for emergency cases. So for that, biosensing technology have a very big future. Okay, it's going to revolutionize our healthcare system. So yeah, this is telemedicine. I think it's already happening now in pandemic, whereby people like they contact their doctors and then they're communicating with their medical personnel using technology. Okay, it's like virtual meetups, just like our meeting here. It's like just virtual meetups. So it's a practice of medicine using technology to deliver care at distance. So again, yeah, it's sensors, health monitoring, and also smart sensors. Why it is important? It is easy, fast, remote monitoring, and saves traveling costs. So next is about career opportunities as a biomedical engineer. So if you are a biomedical engineer or someone from biomedical engineering field and you are an expert in biosensor technology, or you are interested to do biosensor, you are interested to venture into biosensor field, then you can do research. You can become a master's or PhD students. Next, you can become a sales engineer. We need the knowledge of biomedical engineer to become a sales engineer, especially sales engineer means it's for medical devices, okay, to persuade medical doctors, personals in healthcare system. Okay, usually people underestimate or look down on sales engineer because they are doing sales, but the actual thing is they are good at communication skill and they are people with great knowledge in technology, all these medical devices. Okay, so there is an option to become a sales engineer. Thirdly, about maintenance in healthcare industries. For example, if you are working in hospital and you are a biomedical engineer, then you are going to repair all the medical equipments, especially in operation theaters, because I have experience of working as a biomedical engineer during my internship in hospital. And that experience was fascinating because you will feel like you are playing equal role as a doctor in saving people's life. Okay, so in maintenance. And then fourth is science communicator. So this is one of the interesting fields, science communication. You can become a science communicator. You can use this technology. You can spread the knowledge. Okay, you can write blogs. And this is another career opportunity which is yet to be explored. Not many people in this field for biosensor technology. And number 5 is startup. Yes. So I would like to just instill this thought inside y'all. I mean, like when you do a research project, always make sure that you have the idea of commercializing it. It is not for the purpose of making money, but in order to make it like beneficial for others. I think that noble intention must come from us, because when we do big projects with a lot of grant money and then we have patents, it shouldn't stop until research level R&D, but it must be commercialized. Okay, so we must bring it to the next level. So I wish that young graduates will have this mindset whereby they not only being very dedicated in doing your research, but also you must have the strategy together. I mean, talk with your supervisor or your university, have this idea of opening these startups, okay, have this entrepreneurship skill to bring it to the next level so that whatever work you do will be beneficial to mankind. That's the key, is that, okay? And so these are my suggestions for postgraduate students. Let's please look at Publon's Web of Science Academy and also Elsevier Research Academy. There are a lot of free online courses and certifications to upgrade your skills, research skills and knowledge. I only knew about it after I completed my PhD. So that's why I'm sharing to you all, so that it might be helpful to you all to, upgrade your skills, and it is very important. And yeah, lastly, so every dream can become a reality if you have the courage to pursue it. So keep chasing your dream, work hard to it, have a noble intention. I wish you all the very best. So thank you. Thank you very much. Shukran.
Thank you so much, Dr. Rina. Anyone have a question? He can open his mic. سؤال ممكن يقدر يفتح المايك من عنده.
Okay.
Okay, thank you so much. The third speaker is Nibras. Nibras, the mic is yours.
Okay, hello everyone.
Hello.
Can you hear me clearly?
Yes, yes.
Okay, I just would like to thank everyone here, and I'm honored to be part of this session, Introduction to Biomedical Engineering. My part is a general view about digital signals and image processing from the biomedical engineering perspective. So the main point will be digital image processing and digital signal processing, and we will go through some research example. So at the start with the digital signal processing, which is abbreviation DSP. Before that, we can see what is the signal. Signal is a function or data set representing a physical quantity or variable. Usually the signal contains information about the behavior of the physical phenomena. We can also represent it mathematically using a function. For example, if it's a function of time, we can say our signal is x of t. For us as a human, we generally deal with the signals as analog signals. But nowadays, because we need to manipulate and process these signals, we need to use some processing system like the computers. But we know that the computer can't deal directly with the analog signals. So we need to change those signals from analog to digital signal. Here comes the part of digital signal processing, which involve developing different kind of algorithms that we can use it to enhance our signals or extract some useful information. For example, if we have some kind of audio signals and we need to process it, we need to convert these signals from the analog shape to digital so that the computer can understand it. We can do that using what we call them ADC, which is the analog to digital converters. So it changes the analog signal to digital signal so the computer can understand it and process it and deal with it. After that, we need to have the signal in an analog shape so that the human can understand it. So again, we need to use a DAC, which is the digital to analog converter. So it do the opposite, change the digital signals to analog signal so that the human can understand it easily. So for our part in biomedical engineering, the signal we deal with, they call biosignals. So let's see what are those biosignals. Biosignals is any signal in living being that can be continually measured and monitored. So the term biosignals is referred to all the bioelectric signals, and it also includes the electric and non-electrical signals. In our body, there is a various kind of biosignals can be collected by different body organs. For example, we have the electroencephalogram, EEG, which signals come from the brain. We have the electrocardiogram from the heart. We have the electromyogram from the muscles. We have the electrooculogram from the eyes, the retina. We have the ERG. We have the electrogastrograms. And finally, we have the GSR, which is the galvanic skin response. So as I say, we have different kind of signals. They can be occurred from the human body. So some of those signals, they have standard shape. For example, we have like here, this is the ECG signals, and here this is the EOG signal, here EEG signal from the brain, and here is the EMG signals. So most of the signals, they have standard shapes, and some of them, sometimes they vary. So because all these signals, they can have different voltage. So that means different intensity value. So we notice that some signals, they have intensity higher than the other. For example, usually the EEG signals, they have a low voltage, so that we notice the intensity of their signals or the peak of their signals. Mostly they are weaker than the ECG signals. For the second part, which is also the non-electrical signals, they also are biosignals. We have kind of non-electrical signals from our body, like the mechanical signals, the mechanomyograms, EMG. Also, we have the acoustic signals, like the photonic and non-photonics, which we can get them from human precincts. Also, we have the signal, the chemical signals. For example, we can use them to measure the pH and the oxygenation. Lastly, we have the optical signals. So all those are different kind of signals. We can have them from our body. So generally, to deal with those biosignals, we need to first collect those signals from the specific regions, right? So after that, to do this acquisition, to get the information, sometimes we use kind of what we call it transducer and sensors. As I mentioned before, you need some kind of device that can be used to collect this kind of signals from the body so that you can later process it. So we need use that sensor or transducer to collect the physical signals. And after that, we need to put the signals in a shape or in a condition that we can deal with it. And as we say, to process the signal, we need to use some processing system like the computers. But we agree that the computers, they can't deal with the analog signals. So sometimes we need, or mainly we need to use the analog to digital converter to convert these biosignals, these analog signals to digital signal so that we can deal with them, so we can process them. This is for the first part. For the second part, for the digital image processing, DIP, generally this, the digital image processing, some people agree that they are also part from digital signal processing. So we can say that the bigger field is the digital signal processing, but mostly some people prefer to deal with the digital signal processing separately from digital image processing. So let's see what is the image. Image, it can be defined as two-dimensional function, f of x of y, where x and y, they are the spatial coordinates, and the amplitude of our functions, it's represents the intensity in the image. So our image has different intensity. So each intensity has different value. So when we have this function f x of y, we call our image a digital image. So we can say that the digital image is a two-dimensional array, specifically, usually it's arranged in a row and column like this. So this is how it's our digital image can be represented. The digital image, it's contain finite number of elements. Each element have some information depending on its value in its location in the image. We refer to all these elements by the picture elements or the image elements. So briefly, we call them pixels. So the pixels are most widely used to denote the element that represents the digital image. So this here, we have digital image, each point represents pixel. For the digital image processing, it mainly includes the following step to like get the information from our image. We need first to import this image using different kind of acquisition tools. After that, we need to analyze or manipulate our image so that we get the output result. And we need to remember that after processing, we need to analyze it, because sometimes even the processing, it comes in a way it's hard for the human to understand. So after we process our image, sometimes we need another step, which is analyzing the this result to have our final report or final result. Okay, we know that there is generally the electromagnetic field have like different kind of regions. It's also have different kind of wavelengths. These wavelengths have different effects on the body when it exports to it. So in the medical field, we try to use different kind of modalities to help us study and understand how our body works. Where there is like if there is the issue or something like that, those techniques, they help us to know what is the issue so that we can try to solve it. So generally, there is two main kinds of radiation. They are the ionizing and non-ionizing radiation. The main difference between them, the ionizing and non-ionizing radiation, that the ionizing radiation, in it, we have the enough energy to ionise the atoms. It's like in the case of the modalities of X-ray, gamma ray, and the beta and alpha particles. In the other hand, we have the non-ionizing. In that case, we don't have they don't have the energy to ionize the atoms. We find this kind of radiation in sources like the light, the laser, the microwave, and in radar. So generally, the ionizing radiation can be used in modalities like X-ray, mammography, CT scan, and a lot of like famous modalities. And the non-ionizing, like generally in the MRI, ultrasound, and so on. Well, as we can see here, each wavelength range can be used in a different kind of modality, as I said before. So the imaging is generally not just for the disease detection. Sometimes it's we can use it as a main tool for the therapy. So especially, for example, if we are like doing some kind of surgeries or something like that, we need to have a way so that we can decrease the body invasion. So we need to use some kind of non-invasive techniques. So imaging will help us to do that. So a lot of these techniques, they are using for the detection and they are also using for therapy. So after we get all our medical image, here this is a kind of different kind of medical image. They come from different kind of modalities. Here we have like X-ray, CT. Here some of them they like gamma ray. We have another X-ray and so on. So each kind of modalities, they have a specific kind of medical image, but they all like some of them they focus on imaging a specific part, and some of them help us to get specific informations. Some of them, for example, just give us the shape. Some of them like tell us about the function of the organs. So you like selecting the specific modality, it depends sometime on the application that you want to use it in. So to process our image after we get the image, as I say, we can process or manipulate it using different kind of techniques. They need to implement to get like some information from those captured image. For example, we can have like some of the digital image processing techniques. For example, we can do enhancement techniques. Enhancements, we can say generally it include like improving the image quality by like doing some maybe contrast enhancement, spatial filtering, and so on. We can also detect the object from any image using what we call it object recognition. Also, we have segmentations. Here, in the segmentation, we can divide the image into regions, depending on the characteristic. For example, to identify the boundary, to extract specific informations for a specific part of our medical image. Then we can use also some maybe mathematical morphological operation. Those processes, they can be done to our image, and they mainly work on the shape of the image. So the main operations, we have like 4 main operations. They are the dilation, the erosion, opening, and closing. So we can use the specific operation depend on what we want to do on our image. Usually the morphological operation, they use a post process. So after we get the final result, sometimes you need to like do some post processing to get a better result so that we can use some of these morphological operations. Also, we have we can use some representing and description process, because, for example, even the segmented object, they need to be characterized for some like computer processing, like in the pattern recognitions. So we need to represent it or describe it in a specific way. Also, one of the imaging processing techniques, we can use what we call it a color image processing. Generally, when we deal with the image in the medical field, we prefer to deal with it when it's in a grayscale. But also some software, they can deal with the image where they are color imaging. This is the first thing. The second thing, sometime after you process your image, it's easier to represent those final result in a different colors. So it's easier for the person to interpret. For example, if I do the processing, I already understand most of the details. But sometimes after I did the processing, maybe I will get the final result for a doctor or someone who's not familiar with all these techniques. So when I use some kind of, maybe we call it a pseudo colors or like fake colors, it helps the person to understand. It's mainly used on the medical image to help the doctor understand like a specific part or differentiate between some segmented parts. So they mainly, sometimes they are not like from the main image. We can insert them by ourselves using one of the image color like processing techniques. Here we have different kind of software. Most of them are available and used by a lot of people. We can use all these techniques for the image processing, and also we can use them for the signal processing. As I say, they are available and can be used. Some of them, like maybe they mainly focus on like working on specific modalities, but some of them like they can work on different kind of image. For example, for me, I'm familiar with MathWork, which is a MATLAB. So it can be used for different kind of like signal processing and image processing. Some of these software, they focus on a specific kind of images, like the micro diagrams. It's like a specific kind of image. There is also, we have the MEG and Fuji, which also kind of MEG. They are like in the same. So just we have like different kind of software. All of them, they can do the same thing, but sometimes depend on the type of data you are dealing with it, you need to differentiate between them and see what will be like better for you. Because all of them, they have, as we know, they have like a lot of advantage and disadvantage. Even this disadvantage maybe is different between person to person. For example, maybe some software, there is a disadvantage, but for me, it's okay. I don't have problems with that. So I prefer working on MATLAB. Another person prefer using the Python language. So they work on like different kind of the available software. So it depends on your knowledge or depend on your application. So the last part is some research studies. I get here, I have like just 2 examples. Here we have, I think the first one for the signals. The first example is a study titled Diagnosis of Cardiac Disorder in the ECG Signal using the Artificial Neural Network algorithm. In this study, they developed a method for the cardiac arrhythmia detection to differentiate between the normal and disease cases. Their method it's mainly includes 3 main steps. First, they did some pre-processing, which mainly focus on designing some kind of filters, because normally for the ECG signals, it can easily interfere with other kind of signal. For example, when we get the ECG signals from the patient, there is another signal around, we can call them the environment signals. They can all of them overlap or interference with the main signals. So we need some kind of filtering to like remove this kind, what we call it noise. For example, here, this is an original image before using the filter. Here we use some kind of filters. We can use, you can design the filters depend on what you need to do. After, even when you use the filter, you cannot easily get the ideal. For example, this is like ideal result, but it's hard to get. So even if you get this one, it's enough to get information from it, but you try your best to like have maybe the right filter, because sometimes even when you do a lot of filtering, it will be a problem, because in that case you will lose the noise. You will like remove the noise, but at the same time maybe you can lose some of your informations. So you need to take care of when you designing the specific kind of filter you use. So after we filter the signal, then this signal can be dealt with. So this signal here, this is the ECG signal before the filtering, and this is the ECG signal after the filtering. So from these signals, we need to extract some feature from the signals. Here, they use the wavelet transform to extract this information. Because there is hundreds of features they can be extract from one image, we need to have another step, what we call it a feature selection. So first, we just feature extraction. It can extract like a lot of features, but depend on your application. Maybe you are interested in a specific kind of feature which is important for you. You can just focus on them. You don't need to like work on the feature, because sometimes they are just waste of time, and sometimes they affect on your final result. So after feature extraction, we can have feature selection, which is like a specific feature from all the extracts features. For example, for the ECG signals, mainly in the ECG signal, the familiar, I think most of the people try to focus on the like R peak, because as I see, for example, if you see this is ECG signals, we notice that the R peak is a higher one. So it's easier to get these features out, and you can use them to differentiate between, for example, the normal signals and the abnormal signals. Lastly, we need to use all these feature as input to a classifier to classify these signals to like normal or abnormal. Even the abnormal can be classified to a specific kind of disease. The algorithm used in this study, it's artificial neural network algorithm. We can say that briefly, it's one of the famous algorithms that it's work like the human brain. It's need to be trained first, then after that you can test it. It also contain different kind of layers. We have the input layer, the hidden layer, the output layers. This algorithm, I think it's I used it before using MATLAB. There is available tools they can be used there. You can design or you can write by your own algorithm, or you can use the tools that's already available there. So in this study, they were able to classify the signals with the accuracy and specificity of, I think, 98.7%, which we can say it's a good result. So for the second study, for the second example here for the DIP, this is a study titled Detection of CT liver lesions using Adh algorithm and morphological operation. In this study, they aimed to detect the lesion on the CT liver image. They collect their data, which was the real image collected from some local center. They design, here this is the system, acquires the image, do some pre-processing, as we say before, filtering to remove all this kind of noise. They use two kind of filtering, the smoothing filter and the sharpening filter, and differentiate between them to see which one like the performance is better than the other by like optimizing between them. And as we say that, we know that when we have like a CT image, it's usually for the abdomen, not for just the liver. So there is a step of segmented the liver. So it's easier to deal with it. It's better than dealing with like all the abdominal image. So to make it more efficiently, they separate the liver from the other abdominal organs. After that, they implement the Adh algorithm, which is a three-short algorithm to segment the lesions, and they work to enhance the lesion boundary using some morphological operation. Here, this is a part of for the lesions, as I show here in this image. So here, mainly they use segmentation techniques to detect the lesions from the CT livers image. So generally for these topics, I think for the digital signal processing and for the digital image processing, nowadays they are like using in a different areas, not just in a biomedical. We can find them in a like different kind of study, because when you do processing for the image, you can do it for the medical and for the non-medical image. Here we just focus on the medical image because it's like for biomedical image, but these techniques, they can be used for different kind of image in a different field, and you can use any one of like the available software that use there, and they are like available online and they easy to learn, so you can deal with it. So I think that is all. Yeah, I think that's all. So if there are any questions or any comment, they are welcome. Thank you.
Okay, thank you, Nibras. Anyone have question, please? Okay, the last one, الرئيس حقنا ختامه مسك الأخ يوسف. Yusuf, the mic is yours. You can start, please.
Okay, thank you. I think Nibras need to leave the screen.
It's okay now?
Yeah. Well, first of all, I would like to send many thanks to Associate Professor Dr. Sorabong for being here today and contribute to the seminar, for his experience and the transition between two majors, and there are two heavy majors in engineering as well. And thanks to Dr. Rina as well, my colleagues in Malaysia. And thank you for your contribution, and you did a great job, and thanks for being here today. My also colleague Nibras, thank you also for sharing. Well, I will try to not take too long. I guess maybe some of you have feeling sleepy or something like that. Well, it's bioinformatics, trends and developments. It's related to biomedical in somehow, because you can see biosensors, bioengineering, imaging, all these things are in a bio field, and it's somehow related because it's interdisciplinary fields right now, and it's all connect to each other, and working in different majors or different fields can contribute a lot to the science and to the life benefits. Well, as a short introduction, this is my background. I did my bachelor in Malaysia, my master's degree in Thailand, and now doing my PhD in China here. And my research interests are actually in biomechanics with Professor Sorabong, and now I'm doing bioinformatics and biomedical informatics with my current supervisor here, and generally in medical devices. Well, if I would speak in Arabic, I don't know if I speak in Arabic or English, but I think I go with the stream, right? Because it's fully English, so I may proceed with English, and if there are any questions later on, maybe we can answer by both languages. So what is bioinformatics? And you wondering what is it? Actually, it was invented by Pauline and also Penn in 1970s. So it's kind of a new field. Actually, 70s means like almost 50 years right now. But being in the application life or in the medical life, it was like from late 90s, like 30 years ago. So what is bioinformatics is actually biocomputing, application of computers in bioscience. And with the explosion of genomic data now, and you can see a lot of diseases now, and all the genetics, genes, DNA, RNAs, all these are become like data sciences and huge data, and we can like hear a lot of like about big data, huge data, and log data. So there must be something can deal with that and analyze it, and that thing is bioinformatics. It's also accessing and managing the data. It's a very fantastic tool to manage the whole data that we can use. So what bioinformaticians can do or will do, they do or create mathematical models, develop dynamic simulations, and perform pattern analysis of biological systems. Well, as we know, all majors need to have components like biosensor, mechanical or image processing. They all need to have components, and the major components of bioinformatics are actually, or basically the pillars of having bioinformatics in the field. So technology is related, and building algorithms based on mathematical and physics and also calculation methods. Having tools to analyze all these algorithms and using it to analyze the databases or the data sets that we need to obtain from patients, from different species, Homo species, or you can say mouses, animals, plants, and so on. And for all these processes, you need to have a very supercomputers, or you can say good power computing to do all these things. So it's actually heavy things. Well, why bioinformatics? I will not read all that, but stepping it is like molecular, but you have molecular biology data, and you develop a tool to analyze it. After analyzing, you need to make it meaningful and accurate, to explain, to interpret, to give some meaning of it, and then understand the behavior or how the gene can act within the body or human body, and the taxonomy, the evolution of it. And all this comes up with the discovery, or you can say finding an information, hidden information of this biological, you can say genes or DNAs, RNAs, all of that. And that will be used for drug discovery, for medical purposes, and all that stuff. So disciplines that interact with bioinformatics, or you can say the foundation of bioinformatics, is medicine, computer science, math, biology. So bioinformaticians, they cannot work alone. They cannot be alone at all. They have to work with all these aspects, and having touch with these things, you will have a very productive results and meaningful outcome. Well, applying bioinformatics in the lifetime or in our lives, as you can see in the schematic graph, we use it to manage the data, as we say, development of new algorithm. You need to be very good in mathematics, statistics, and so on, analyzing and interpretation of data. Also, we need to develop tools. What are the tools? Like, you are using some tools to solve some like particular questions, particular issues, solving like research questions, whatever. So these tools are important as any other tools in any different majors. But bioinformatics tools are more softer than other places, like programming. You need to do coding or whatever. So it's all working by your hands and fingers on computers and other devices or machines. And then you do producting, or you can say the production of protein structure and DNA structures, RNA structures, and so on. And the outcome, of course, we need to go with health, drug discovery, because we are working on biomedical, biomedicine, bioengineering, and all this related to our health and our safety and so on. As we know that we have now COVID-19 is approaching everywhere and breaking through every borders. So this where comes science as bioinformatics, or scientists in bioinformatics can come with tools to solve these problems. So in BME is actually a short name of biomedical engineering. So it's a promising field. I can tell it's revolutionary now, and it's working or you can say it's becoming too fast track now in the science, solving biological problems, providing solutions to development of biomedical tools, softwares, devices, and so on. And it's also become one of the, you can say one of the most important or keys for cancer discoveries and as many studies, and also for COVID-19, where we will touch on next slides. So research directions. If you are working on bioinformatics, where to go? Actually, it's based on your background and based on your interests as well. So you can go through, or you can go to DNAs, RNAs, and proteins. And if you are working on DNA, you have to know that you need to do to study mutation of these DNAs and determine the effects on genes, because it's all working how the genes act in the human body, how the genes behavior, and all disease or all kind of like infections now we have is about your gene behavior, gene mutations, and how they act inside your human body. And if you work on RNA, you need to study the expression of that genes, how it's defined, how far it's known in the cell or in the human cells. So expression of the RNA sequence. And then the protein, which is examining the unfolding patterns of genes. So why I'm circling actually RNA? Because it's my research field, it's my direction. And if you are like your background is engineering, or you have basics in programming and software, or computer science and whatever, or any softwares, it's better you go for RNA. But if you're coming from like medicine or molecular or other biology or something, I think DNA or protein is your I think space to work on, to study. So I'm touching here more deeper in RNA sequencing. And as I said at the beginning, it started in 1970s. So that were very basic tools, or very basic machines were invented and discovered to sequence the data, the RNAs, and get like samples from the human body and try to express that data into genes and cells and so on, and then identify the condition of that human body or human like samples. And then in 80s start to like get the qPCR, as you know, like now BCR, the BCR test and so on. And actually it goes through a lot of process now and developed very quickly. In 90s, we start to get microarrays chips where we can collect the samples. But in the 20s, in 2000, after that we got RNA, which is more developed machines, which can acquire, or you can say sequence millions of reads for RNA, DNA, and so on, that like in one sample or in one collection or one study can get like more than a million of genes expressed in thousands of cells. And then now we have actually single RNA sequence is actually invented by Professor Tong. He is a Chinese professor in Beijing University, and he is the first one who established this method and technique is single RNA sequence, single cell RNA sequence. What is it actually? RNA and single cell, as you can see here, the juice here and the fruit salad. In RNA sequence, it comes in the bulk cells, so it's difficult to determine, difficult to identify, and difficult also. It's challenging, actually, a challenging technique. But once it comes to single cell, you are working with a very specific cell, and each cell that they have like hundreds of expressed genes, and easily you can get like the type of cell, the type of disease, and the type of behaviors. It's actually causing several diseases, or that cause you the disease. So if you are working in bioinformatics, you need to know where is your sources, right? So there are some examples of these databases, and it's huge right now, actually, with the development of technology, internet, and institutions, and also a lot of fundings in this field. So you have like DNA and protein sequencing if you are working on DNAs, molecular structures, phenotype, and biodiversity. So I'm working on phenotype stuff, which is analyzing the expression of genes. So here are some popular, or you can say open access databases. As you can see here in this scheme. So these are the most popular, or you can say most known databases, and it's actually open access and also public. So it contains millions of databases and hundreds of millions of genes and also expressions. And also, if you want to be specialized, more specialized, you need to go to the that some institutions or different like it's sometimes paid, or you can say it has strict access to some institutions. So bioinformatics as well, you can see how we can get the data or how they do the data. Basically, there are labs everywhere in the world, and these labs sequence data or take samples from human bodies, from different species, and then archive it into libraries. That is called PLAST and FAST, and too many libraries. They archive that, they classify it into different types, like, as we said, human, animals, plants, and so on, and then they share that data with other sources and compare the quality of the data. They do quality control, they do normalization, they do a new sequencing, and then they come up with a clean, good data that can be, because when you have a bad data, or you can say unfavorable data, then you will have bad results, or you can have could be disadvantaged results, and then you are misleading the research as well. And then they analyze it. After that comes to the bioinformatics, which is developing tools to analyze it and make it easier for researchers, for sectors, and so on. So you can see a very example. It's actually an old example, which is between 89 and 1099. In 10 years, how huge the data are increasing, and the databases. This is one database. This is GenBank and NCBI, which is PubMed actually. So you can see here in millions of data. So it's very huge data, and then you need to have very supercomputers, or you can say computing power. In the market, if you want to work in the market, so you need to know what are you going to. As we know now, with COVID hitting everywhere, a lot of industries been struggling and they suffered a lot. But some industries as well, they got a positive impact, and one of them is bioinformatics industry. And I guess it doesn't mean other majors, I mean, like, are not very lucky or something. No, it's, I mean, these, as you can say, if you go to research now, or you can say to go to the sources of research pages or research data set, you will find that bioinformatics is approaching, bioinformatics is leading. And as you can see in this study here, they mentioned that between 2019 and 2027, how can dramatically the bioinformatics market will change all over the world. And here you can see here right now, actually, America and Canada, they are leading countries for bioinformatics. How to trying to fast, as you can see here, like maybe for the last 10 years, as my supervisor said, it's almost like all universities, or most of the universities, big universities, they start to open institute for bioinformatics or lab for bioinformatics, because it become like a policy of the country and policy of science as well. So as you can see, maybe the lamia here is still like, which is Middle East and Africa and Latin America. It's still maybe late for this bioinformatics or this field. We hope that in future we can contribute something to that. So I think this is my last slide. I try to minimize it as much. So thank you so much. And the floor is open for questions and answers.
Thank you so much, Yusuf. Anyone have question?
Yes.
Any
Hello?
questions?
Can I ask a question? Can you hear me?
Yeah, please.
I have one question. Bioinformatics is for this field for me is new, but I want to ask one question about the topic, the hot topic of bioinformatics, the future direction for this, and with telecommunications field topics, like with 5G, with this technology. What do you think? What the future? How we can mix this together and how our.
Well, okay. Actually, you have. If I get your question, you mean like how we relate bioinformatics to the new technology of 5G and of communication or telecommunication technology? Is it?
Yes, with machine learning, with artificial intelligence, machine learning, we know something like with classification, something like that. But with telecommunication, with this field, with IoT, mobile edge computing, these things with different field on telecommunication, we can mix together these two topics.
Well, to the best of my knowledge, as I'm not like a telecommunication engineer, but there is a part of bioinformatics called informatics. If you leave the bio, you become like informatics. So you can say it's actually, when I search about it, you can teleinformatics, health informatics, and other parts of informatics. So informatics is dealing with data, and huge data. Actually, it's dealing with data and the big data right now. And we know that internet, telecommunication, and 5G is actually based on data, and the world is, I mean, is now crazy. It's like maybe the next wars. It will be data or the big data. So it's really, or you can say it's very much correlated. It's very much connected to each other. Informatics, but if you call it bioinformatics, then you need to work with any bio stuff. I mean, any bio-related research. But then you go to informatics as a specific field. You can use it to analyze the data that come from 5G now, from the internet, from the connection, the wires, also a lot of now you can say social medias, and it's all about social media browsing, using internet for like collecting this data, and also for surfing. So it's very much related, and it's also the future of being a scientist or data scientist in the coming future. And but if you ask me in bioinformatics, what is the trending, or what is maybe the hot topic for the next 10 years, I can tell you single cell RNA sequence analysis, because this is like it was started like 10 years ago. And right now, many researchers, I never saw like there's a paper published in a very low impact factor. Mostly are in Nature, in Cell, and it's very hot topic, and it's very emerging. So if you're going to do like bioinformatics or working on that kind of things, I think you should go for single cell, because it become very fantastic, and it's isolated cells, and it's very important if you want to discover something or like disease or like COVID or whatever. I hope I answered your question in somehow.
Yeah, thank you very much. If you after we can contact you by the email or something.
Yeah, right. I think, are you in China?
No, I'm in Russia. So they proposed for me some jobs with bioinformatics, but I finished the PhD from China about telecommunication, about 5G and something like this. So,
I think you know that China is now leading the 5G technology.
Yeah. Yeah, so here in Russia, the 5G is not like in China. They focus on bioinformatics. So I want to discuss with you some.
Pure, you mean pure informatics, not bioinformatics, pure, right?
No, bioinformatics.
All right. Okay.
Discuss about. No, thank you for your answer. We hope to contact you soon.
Sure. Thank you so much.
Thank you.
Okay, Sulaiman Ward. You can ask.
Hello.
Yeah.
Hello.
Hello. Can you hear me?
Yeah.
I just want to say that thank you a lot for professor and doctors for this interesting lecture. And I just want to ask Professor Sorobon, if I
Yes.
pronounce the name right. What if I want to ask about contact?
Okay. I can give you in the chat box, I think, if like that.
Mm-hmm. Okay, thank you a lot.
Thailand is a beautiful country. If anyone wants to go to Thailand, it's a very nice country, and you will like the food actually.
Thanks a lot.
Thank you, Professor. Thank you.
Yusuf, can I ask you questions?
Yes, yeah.
Yeah, because it's really great that I hear the talk that you talk about the bioinformatics. Like as you may know that you come from mechanical engineering also, right? And become like a biomedical engineering.
Right.
Yeah, and now you are bioinformatics. Very interesting. So as you mentioned that about the technology on the single cell analysis, I just wonder like anything that can link in the future for the treatment, like a medical treatment from the single cell analysis, in your opinions?
Yeah, actually, the novelty of single cell is it's very specific to any disease. Let's say if we work on aneurysm, like the work we did before, and there's a new paper is published in I think beginning of 21. So using single cell technique for dealing with specific disease, it's more convenient to understand the gene behavior in a specific cell, because let's say you are working in blood cells, so you need the specific cells of the blood. So the single cell, it will give you the specific genes, and you will know that specific genes for each cell. And once you track the genes, or you understand the gene behavior, the gene attitudes inside the cell, you will have easy tracking for the expression, for the analyzing, and you will understand whether this is the diseased gene or is the healthy gene. That is the technique, because it gives you, as like I show in the fruit salad, you will know that this is the targeted gene, and then you will look at that gene specifically, and it will be your aim for the treatment. Then you need to isolate the gene, or you need to have find whatever it takes to make that gene works again or be healthy again. That is actually the scenario, or you can say the novelty of single cell RNA sequencing techniques. So if I get much better, I mean, it's failed and the more deeper, maybe we can have a collaborated research like cardiovascular and also the single cell, because it's really approaching and it can interfere anywhere.
Yep, that's a great idea. Yeah.
Maybe biosensor too.
Okay. Another one have question. Anyone, please? Okay. Professor Surabon, Dr. Arina, Nibraz, Yusuf, thank you so much. And we hope to see you with us at future seminars. Thank you again.
Yeah.
Yep. Thanks.
Thank you. You are welcome.
You're welcome.
Thank you, Professor.
Thank you all.
Thank you.
Good night.
Thanks.
Good night.
Yeah, good night.
Take care. Good night.
Have you a good day from here, from Russia?
Okay, thank
Morning there, right? It's morning in Russia?
No, it's evening now. It's almost 4.
Okay, nice to meet you, man.
Yeah, I will search your email and contact you, please.
I can send you in the chat. I can send you in the chat.
Okay, okay.
Do you have
thank
WeChat?
you.
Do you have WeChat?
No, we have this big lab here, but the knowledge here in Russia,
Yusuf, you can send your email in chat now.
Okay, okay.
I will
thank you.
send it. Okay. Yeah. Okay. Thank you so much. Thank you so much.
Muhammad, you did?
Yes, I got this email.
Okay, bye-bye.
Okay, have you a good night.
Bye-bye.